All right, we have 1 minute. Holy moly.
This is good. This is what you want.
Yeah. There's 2 seats up here. Cool. Yeah, Sandy and Rick texted me earlier, so. Hey, how's it going? You're so close. I might like trip and fall on you. This is going to be, yeah. Yeah.
All right. It's 4 o'clock and it looks like everybody is settled in. Thank you so much for showing up to this special time seminar. It is my great pleasure to introduce to you Ben Perlman, who has been a graduate student in my lab the last 5 years. Ben got his bachelor's at Uncle Charlie's summer camp, also known as University of California, Santa Cruz, which happens to be my husband's alma mater, which is kind of weird. And then he did his master's degree with one of my lab mates from graduate school, Laura Ferry, at Moss Landing Marine Lab. And then he came to us, and most of you know Ben pretty well. He's made a mark in the department. Not only has he been incredibly enthusiastic in getting his own research pursued, but he's directed numerous undergraduates. He's helped develop an entire new course centered around Belize, which has been wonderfully successful. And I'm kind of thinking, is there some way that I can flunk him so that I can keep him around right now? But, I guess not. So we're going to have to let him tell you about his dissertation research, biomechanics of an odd little fish that spends time on land, kinematics, kinetics, and motor control of the mangrove rivulets. Take it away,
cool.
Ben.
All right, thank you so much for coming. It's nice to see all your faces, especially a lot of people from my spring Belize class just trickling out of the woodworks. If you don't mind turning all the lights off, hopefully nobody will fall asleep. I am trying to convince you that this fish is worthy of your time and darkness. So let's go ahead and get started. So it is a central tenet in comparative functional morphology that form follows function. So to elicit our first example, we have this beautiful albatross with these long, slender wings, which allow it to glide over entire ocean basins. We also have the cheetah, which is able to stealthily chase potential prey items through the open savanna with the ability via its extendable hind limbs. It has a very flexible vertebral column and a very streamlined body profile. We also have tuna that also have a streamlined body shape along with this deeply lunate tail, allowing it to cruise through pelagic waters. The arboreal nature of this chameleon is possible because of its modified forelimb and hind limbs, allowing it to easily grasp tree limbs and branches as it moves throughout the forest. This kangaroo, which is a saltatorial jumping, hopping champion, is possible based on the spring-like tendons in its hind limb, allowing it to move very efficiently through these open grasslands. So in these 5 examples, these animals are specialized to move in a particular environment. Now, what happens if an animal occupies 2 extremely distinct environments, such as the mud skipper and amphibious fish? So they have modified pectoral fins, which are extremely strong, stout appendages, allowing it to crutch along these muddy substrates. So we have this expectation that anatomical specialization will provide evidence for what the animal actually does. So we see this expectation in this artist's rendition of these early tetrapods with these stubby little pectoral fins which evolved in the water, which eventually gave rise to limbs for terrestrial locomotion. Now, these major functional transitions are often accompanied by morphological changes. So here we have Tiktaalik rosier. It's an early tetrapodomorph fish from the late Devonian, about 365 million years ago. Now we have this fossil example on the left with the artist's reconstruction on the right, and I want to point out their pectoral fins, which have been modified along with fin rays that have bendable elbows, a very flexible wrist, allowing them to prop themselves off the ground. They also have a neck joint, which allows the head to move independently of the body for more efficient locomotion out of the water. So what happens if these morphological, these functional transitions are not accompanied by a morphological change? So if you take this fish, for example, the mangrove rivulus, Cryptolebius marmoratus, and throughout the rest of the talk I'll most likely refer to this as a Kmar for short, would you be able to suspect that this fish was anything but fully aquatic based on its external morphology? So what's really interesting about this particular fish is its ability to move on land. So this is one of the first videos ever captured of these fish, and they're actually jumping out of a log. Now, what I want you to really focus on here is the fact that they are directing their movement away from this person's hand to efficiently get away and escape this potential predator. So let's learn a little bit more about the background of this fish. They are a quasi-amphibious teleost, a bony fish that makes occasional forays onto land. They're found from central Florida down to southern Brazil in the Atlantic, along with the Caribbean islands. Now, this is the general orientation the fish takes in, that it occupies in water. So unlike the late Devonian Tiktaalik example, which has obvious external morphological features, this fish lacks any obvious external morphological characteristic, suggesting that it would be able to efficiently move from water to land, let alone coordinate its behavior out of the water. So let's focus on some of its fins. They have a streamlined body shape, and here's its dorsal fin, which is set far back along the body, directly opposite its anal fin. They have a nice lobed plate-like tail fin, the caudal fin. They have their plate-like pectoral fins up by the cranium region. Now, this picture on the bottom right is a fish in its general orientation when it's out of the water. We took this in the field. Here's its head over on the left, and its tail curved on the right, sitting on this nice little leaf. So this species has become an important model organism, mostly because of its reproductive strategies. They are one of only 2 known vertebrates to science to be a self-fertilizing hermaphrodite. So they're simultaneously a male and a female via their ovotestis. They have sexual fertilization, which occurs internally. They lay their eggs externally via oviparity. Their offspring are technically isogenic clones because they share the same genetic material as their parent. And one thing that's really kind of bizarre, they are also males. There's no females known in their population, so if the eggs are raised in cold temperatures, they will be born a male. They could be born a hermaphrodite, and then they could basically stay in cold water for an extended period of time and then shift to a male, which is known as a secondary male. And they are not known to shift back to a hermaphrodite. So each individual will lay an egg and can eventually, via multiple generations, give rise to an entire population that you see here. Some of the reasons these fish leave their aquatic habitat are due to poor water quality. They could be searching out a prey item such as other fish, mosquito larvae, and insects like crickets and spiders. They're also known to escape predators such as snakes, birds, and fishes. This Kmar over here didn't get the memo because this mangrove water snake, Nerodia clarkii, doesn't really seem to care about its prey item right here, and he doesn't seem to be very afraid of this particular snake, which is really weird. The snake eventually ate that fish. So one of their interesting adaptations for remaining out of water, and there was a study done by Scott Taylor in 1990 that recorded these fish to remain out of the water for 66 days, so just over 2 months. Now here is a picture of their gill structure. In the water, you'll see their secondary lamellae with all this space in between. Now, when they've been immersed out of the water for 24 hours, you can see that the interlamellar cell mass has completely filled in. So they're able to reconstruct their gill filaments to decrease the opportunity of desiccation and collapse of their gill structures.